Sandra Hewett was recently inducted as the inaugural Beverly Petterson Bishop Professor in Neuroscience at Syracuse University. Hewett comes to Syracuse from the University of Connecticut, where she was involved in the Neuroscience graduate program.
Neuroscience is an emerging area of excellence now at Syracuse University, and the Bishop Professorship is allowing me to spearhead that neuroscience initiative. And so as a scientist, I'm very excited about being in a wonderful new lab environment here at the Life Sciences Complex, which is absolutely gorgeous. And this gave me an opportunity to build something and come here, work with the wonderful faculty that are already here in neuroscience, but also to hire new faculty in areas to complement those that are already here and to shore up some weaknesses of where we need to bring in some more people so that we will have a great group that can actually work together and hopefully make new discoveries. My laboratory is really interested in the process by which cells die in the brain following cerebral ischemia. And cerebral ischemia is what's commonly known as stroke. And it's really important because, as you can see from the slide, it's the most common neurological illness in the United States. It's the leading cause of adult disability, the third leading cause of death, and there are no neuroprotective therapies. And what that means is there's nothing that can preserve brain tissue once there is a clot that blocks an artery. There's an area here in yellow that is known as the penumbra, which I like to call the walking wounded. And I call this the walking wounded because those cells are either destined to die, as you can see in this progression, and they become red or infarcted, or if we could understand what's going on in that area, we could devise a therapy to block that progression and preserve that brain tissue. Two things that we really think about a lot are excitotoxicity, which is when cells literally excite themselves to death because their cells release their neurotransmitter and these other wonderful cells can't take it back up again, and inflammation. We study the process of cellular death that occurs using two different models. The cell culture model, so we can model stroke, we call this stroke in a dish, and look at the interplay between these astrocytes and neurons and what occurs when there's now simulated stroke or oxygen glucose deprivation. And additionally, we can use an animal model, and we're able to look at what happens in a mouse in this case, which actually can be used as a pretty good model for human stroke, to look at eventually the cell death that occurs in the brain. In science, you go where the questions take you. So I'm not always sure where I'm going to go next, but what I do know is what my next experiment will be. And based on the results of those experiments, sometimes you just take a very different turn.